Sorry for the delay, a little bit technical glitch to get this set. So I welcome you all for this wonderful interaction which you all are going to have with a very, very distinguished scientist today. And so I welcome on behalf of Founder President, Dr. Ashok Chauhan, the Chancellor, Dr. Atul Chauhan, and all members of Amity Fraternity. I extend a very warm welcome to one of the distinguished biochemists, scientists, and an institution builder, and a great person who has contributed to biomedical research in the country, Dr. Alok Dhawan. Let's give a big clap to Dr. Alok Dhawan. I know his father for a long time, Dr. B.N. Dhawan. Many of you of my lineage, my category of age will know he was an icon in the field of drug development. CDRA means B.N. Dhawan. That's the kind of image Dr. B.N. Dhawan enjoyed. So from that time onwards, I have known him and I have seen him growing, Dr. Alok Dhawan. But then he made his own the professional career, not depending on the shoulders of his father, but he built his own the capabilities, image, contributions, and made an impact on his own. So Dr. Alok Dhawan, he is now currently director of the Center for Biomedical Research, which is an autonomous organization under the UP government. And he is at the apex level in terms of his position as the director. And in the level, he says it's level 17. That means almost equivalent to the chief secretary, the state level. So he is occupying such a very important position right now as the Center for Biomedical Research. The organization which he has built in a big way, he brought a new paradigm there by establishing facilities, building the infrastructure, like one of the largest capacity NMR is available over there. Similarly, imaging facilities available there. And so he brought new paradigm in that institute, and he is PhD from University of Lucknow in biochemistry. And subsequently, he also got DSc, Doctor of Science, honoris causa, from Bradford University in UK. And he has a lot of connectivity still with United Kingdom because of the Bradford connectivity. He still enjoys a very good reputation in UK as well as in the institutions, universities over there. Then he is also fellow of many national societies, including National Academy of Science, National Academy of Medical Sciences, and also the Neuroscience, and many, many other society, including fellow of Royal Society of Chemistry. So he is highly accomplished in terms of the educational background. Then he is a visiting scholar in many universities, but I would mention he has been at Michigan State University from his younger days. He has been associated with Michigan State University. He went on the postdoctoral fellowship of DST Bioscars Fellowship to UK, University of Surrey. So he has such a credentials. If you look at, I have circulated his profile. All of you would have seen it. So such a great accomplished person. And when you look at his professional career, the journey of the responsibilities we have shouldered, he has been an institution builder. Wherever he has gone, wherever he has been serving, he has really brought a new paradigm there. Starting with, I'll go backwards. Currently, the Center for Biomedical Research, but then that is in Lucknow. And he has been also in Lucknow at IITR. He has been the director for 5 years, more than 5 years. So he was at the Indian Institute of Toxicological Research. Earlier it was known as ITRC. Then is a CSIR distinguished apex organization. I think you also have interaction with IITR, Dr. Nujindal. And so he has also been director of CDRI, Central Drug Research Institute, which Dr. B.N. Dhawan was there almost for 15 years. I think he was director there, long years. So now he has also been director at CDRI Lucknow. And he was the founder director in Institute of Life Sciences in Ahmedabad University. It's a private university, Ahmedabad University, but he has been the founder director of Institute of Life Sciences. He brought a new concept which I was just discussing with him. At some of the major facilities you can build if you win over the companies like fax machine. He has the company owned, but you operate it and you use it. That kind of concept. So investment, least investment, but company invests, but you give other visibility in their publications and others. So maybe that we can look at this model in some of the institution where you want to build, negotiate with the company, and then bring that infrastructure here at their cost, company's cost, but then they amortize the cost of investment by giving other services to outside, earning money, and then compensating, putting a technician there, maintaining the equipment. You only give the space and the electricity and support facilities. So this kind of concept he has brought in the institution where in Ahmedabad University, that's also a great model which we can emulate as well. He has been also the institution in the Assam University. He served in Assam University, that is in the Department of Biotechnology in Silchar. There is a very important laboratory there. So again, he brought new vibrance there. He brought two important area, animal biotechnology, because India is not only we own a large biological wealth, human, plant, also animals. All kinds of animals we have, starting from cows, sheep, goats, and so many hooved animals, and also the ponies and so on. So how do you take care of this animal health? So he has worked in that Silchar, Assam, and also he contributed to Northeast development in a big way. And he built the Biotechnology Industry Connect in the Department of Biotechnology. He brought the Industry Connect, and also the other dimension which I enjoyed looking at his profile is the student research. He focused on student research, undergraduate, postgraduate, doctoral students. Not only faculty research, you should harness that in a big way, which he has set model. So I was very happy to see the augmentation of student research wherever he has gone. We should also look at that in a big way, and we are doing it already. But then, how do we also see adopt some aspects of best practices he used in terms of bringing student research? When I looked at his some of the national missions which he launched, that was fascinating. When he was in IITR, IITR was known for the toxicological research. So he brought the food safety, which is very important. Because whenever you do post harvest, convert them as a value added product, and how do you bring the food safety and then consumer safety? So this is, he started a national mission with multi institutional participation. So he launched. So this is very important that we have a leader who can look at a national mission. And then he also brought food and industrial toxicology. And we all use nanomaterials. We do a lot of research in nanomaterials, but we should be concerned about its impact on the environment, because ultimately you are going to throw this nanomaterial to the environment. Or some of them you may be using, consuming, or you may be inhaling when you are doing research. So, what is the safety as well as toxicity of this nanomaterial which we all produce, and then some of them are now in industrial scale being used? So, we should look at, and he has developed that facility to test the safety of the nanomaterials research. Environmental health he has contributed. One important product, he has brought many products, but one which fascinated me was the O-Neer. O-Neer. Neer means water. O-Neer. That is free radical, oxygen free radical. Once you generate, this can go and kill the bacteria in the water. Microbial contamination, which is the major problem for waterborne diseases. So he has developed an instrument which has gone to the market. Industry has commercialized this. So using this oxygen, can you kill the free radical of oxygen? Can you give the microbial contamination in water? So you need to only, the water has to pass through this, the free radical oxygen, and then kill the microbes. So its water becomes potable. So this is, I'm just giving you only a glimpse of his achievements, because if I have to read his biodata, it's huge. So we are having such a wonderful person today, and have just given a little tip of the iceberg of his accomplishments. And the topic in which he is going to speak is very close to everyone's heart here, particularly founder president, Dr. Ashok Chauhan, who for him, innovation is his life. Breath, yeah, life is innovation. So he is creating Amity as a cradle of innovation. Because every thinking, pedagogy, the syllabus, curriculum design, everything is looking at how do we transform Amity universities and institutions as cradle of innovation. Because even our schools, we promote innovation at school level itself. And there are many successes coming from the innovations of our school students. So that is the culture which Dr. Chauhan, Dr. Mrs. Amita Chauhan, the chancellors, and all of us in this mission of Amity Group, we are in that process. So the topic is related to that. The topic is supporting risk in science, technology, and innovation for Atmanirbhar Bharat. Honorable Prime Minister has really, in the last 10 years, remarkable change has come, which is palpable, perceptible, and everyone feels, and the whole globe has appreciated Atmanirbhar Bharat. So this mission in defense, you see so many, the 3 lakh crores of system being inducted into the services from DRDO developed items manufactured by defense industry in defense sector. Similarly, in every sector, now health technology, a big mission has been launched. So there is a lot of efforts, Atmanirbhar Bharat. Not only that, make for the world. Why do you stop with Atmanirbhar Bharat? Make for the world. So that is what is the challenge today. And how do we gear up in the Amrit Kal next 25 years? And how do we make our country number one in the world? Not just only number 3, number 4, number 1 in the world, which founder president has been mentioning, envisioning for decades, even 2 decades before. He said that India will be a global superpower. So the topic is supporting risk in science, technology, and innovation for Atmanirbhar Bharat. So what is the risk in this domain? And how do we support? How do we combat? How do we face these challenges which will be coming in the way of Atmanirbhar Bharat through science, technology, and innovation? So that's the topic which Dr. Alok Dhawan is going to share with us. Please feel free to interact with him, because he is here. So it is that every one of us can give some ideas, thoughts for him, and we can also share his thought with us. And ultimately, we adapt them in our ecosystem, Amity ecosystem, and then we will take it forward. So with this, on behalf of every one of you from here, I extend a very warm welcome to Dr. Alok Dhawan. And since he is a part of CSIR, and many of us know him, right from Dr. our IHBT director, who is sitting here now, director of our Amit Institute of Biotechnology, Dr. Sanjay Kumarji. Then we have Dr. Chakravarti, and also Dr. Neeraj Sharma, because of his connectivity with DST, he knows him. And Dr. A.K. Singh has closely worked from DRDO in many projects and things like that. So every one of us know you, Dr. Alok. So we are all very eagerly looking forward to hear from you. And Dr. Chauhan has sent his greetings to you and best wishes for you and for your family. With this, thank you so much. I now present Dr. Alok Dhawan.
So very good morning to all of you, and thank you very much, Dr. Selvamurthy, for this rather generous introduction. My friends on the dais, very dear friends, Dr. Sanjay Kumar, Dr. A.K. Singh, Dr. Chakravarti, Dr. Gopal Bhushan, uh, members of the faculty of Amity, and of course my dear students who are at the back, but I hope they are all awake. Uh, I think it gives me a great sense of pride having seen Amity grow. I've been in touch with Amity for about 23 years, perhaps maybe more. And I've seen how they've grown from strength to strength, not only in terms of the infrastructure, but the kind of thought process that is coming into Amity in terms of innovation, in terms of research, in terms of how students should get the or derive the maximum benefit from the system. So I think it's fantastic. And now that I hear about these Amity International Schools and other schools, I think it's, I'll just add one more thing, sir, before I begin. We experimented in CSIR way back, I think in 2016 when I joined, to bring in school kids to do research. Okay? That later on became a program of CSIR called Jigyasa. And you could actually bring them right from class 10 onwards or 9th onwards. They'll spend 4 to 6 weeks in your lab. They could actually be part of the research program that you're doing. So I think since Amity is integrated through and through, it might be really worth to bring students who are really bright in school, or I would say creative in school. They may not be bright as per the teacher's perspective, but they're creative in school. I think teachers look at marks, but other than that, and the other thing I would mention that as far as what Dr. Selvamurthy said, I think it's very important that we look at the model of minimum input and maximum output. I think that is what this country requires. How we can rope in various stakeholders who are part of the education system, higher education system. And it's not only in India, but I think globally, there's the challenge how to make everyone partner for this global education system that we are now getting into, especially with the new education policy. How we can rope in large number of student population into our research programs. And they would be like the bottom of the pyramid, if I may say so. And then you move up the ladder. The students are groomed. They get plenty of time to do research during their curriculum. So I think that's how things are moving forward. As far as supporting risk in science, technology, innovation is concerned, I think I must openly say I'm a major beneficiary of that, and that is why I give this title. Why I say this, that I think even before the concept of Atmanirbhar Bharat came in, or the concept of the fact that we'll be a developed world in the next 25 years, Amrit Kal came in. A large body of work was being supported in terms of risk. And I think that is how institutions have been built in this country. But I think now it is more institutionalized in terms of the fact that if you are willing to take a risk, there's always a body which will be there to support you. So I thought it would be a good idea to share some of my experiences with this August gathering. And some of the slides, because I was told there will be students, which there are. So I think the faculty have to excuse me if they find them a bit under the par. So as Dr. Selvamurthy mentioned, I have been part of the CSR network as well as now with the government of UP. So I'll be representing both the aspects. So this is the institute currently that I head. It's called the Center of Biomedical Research. It has now been approved to become an institute of national importance, which will be the National Institute of Translational Research. The land has already been allocated. We are now looking forward to the cabinet approval for moving forward. So before the other thing I always mention, unless you have something to look forward to, I think it's very difficult for everyone to get motivated. So when Dr. Selvamurthy asked me what was the vision of this institution, so I just said it's advancing frontiers of science for human health and wellness. And this is a broad tagline, and I believe in this very strongly, that unless you have a tagline that people look forward to, it becomes very difficult for you to sort of rope in people from different walks of life. So this was a tagline that I provided to the institute after joining, and which is now copyrighted to the institution. And it's similar to institutions as well as products. When you look around, I think if there's something that lingers in your mind for long, it's very difficult to remember the vision and mission of an institution. But if you have a tagline, that is always remembered. The other thing why I always put it up in the front is the fact that anything that you do, you should protect the IPR. That's very, very important. That's an intellectual property that you have created. So you should try and protect that as much as possible. And I'm very glad that the honorable founder of Amity University actually believes in creating a lot of IPR. And I think among the private universities, I think Amity is on the top in terms of IPR. So that's remarkable. What I'll do for this, to begin with, for the institution, I thought rather than spend time, I'll just play a small video so you can have a sense. Is sound? This is my sound. Center of the government and it's Department of Medical Education. The vision of CBMR is to undertake, aid, promote, develop, guide, and coordinate basic clinical and translation research. And under this vision, the mission of CBMR is to establish an enabling ecosystem for cutting-edge interdisciplinary research and entrepreneurship leading to better patient care. This center was started as a Center of Biomedical Magnetic Resonance. And in the year 2013, we enhanced our vision to make it the Center of Biomedical Research. Center has been working in multidisciplinary areas to undertake cutting-edge research in biological and chemical sciences. The center, over the period of almost 17 years, has been relentlessly working in the area of better patient care using clinical metabolomics as well as functional MRI. It has not only helped the clinicians in the hospitals to be able to identify the disease conditions, but also to be able to track and monitor the therapeutics of the diseases that they are following. CBMR has been closely associated with institutions such as the Sanjay Gandhi Postgraduate Institute of Medical Sciences, the King George Medical University, and the Ram Manohar Lohia Institute of Medical Sciences. Apart from that, we have been now associated with various institutions, hospitals, universities, IITs to be able to exploit the full potential of this center. To undertake cutting-edge research, CBMR is equipped with state-of-the-art facilities, both in the area of biomedical sciences as well as in chemistry. Over the years, the Center of Biomedical Research has emerged as a premier research institution in India in the area of disease research. We are now being able to develop biomedical devices which are capable of identifying or differentiating disease conditions using the biomarkers of NMR. For this, the center has actively collaborated with IIT Mumbai. And once these devices are ready, we will be able to serve the primary health centers. This center is very unique as it has a 800 megahertz solution state NMR, which is utilized for clinical metabolomics. It is the only one of its kind in this country. We also have a 600 megahertz solid state NMR, which can be utilized for cartilage and bone. And again, this is the only one of its kind in the country. And we are actively collaborating with international universities for these kind of researches. Using the 3 Tesla functional MRI machine, we have been able to probe the various cognitive dysfunctions or deficits that are observed, not only in children, but also in adult population. This has now helped the clinicians in identifying these at a very early stage. Some of the examples, of course, are ADHD and bipolar diseases. The center is advancing frontiers of science for human health and wellness. And keeping this in mind, it is proposed that we will soon be establishing this as an institution for translational research. The center has also contributed towards very high-end synthetic chemistry, which will now be very helpful in the area of drug development. The center, with an ambition to become an institute, has established different departments such as the advanced spectroscopy and imaging, systems biology, data sciences, biological and synthetic chemistry, biomedical engineering devices, and of course an incubation cell to make this center, and not only center, Uttar Pradesh, an Atmanirbhar state. Through this, we'll be contributing towards the various mission programs of the country as enshrined by the Honorable Prime Minister, and of course the state as enshrined by the Honorable Chief Minister. The center supports research in the area of medical and functional MRI, biomolecular NMR, molecular synthesis, translational cell biology, and chemical biology. The center produces high quality research, and the Nature Index in 2020-2021, when it assessed the government institution across the world, CBMR was assessed at number 4 in chemistry in the country and among top 10 in all subjects in India, and was also ranked among top 50 in the world in chemistry and among top 200 in the world in all subjects. This only goes to show the high quality research being undertaken at the center, for which I am indeed grateful to the Scientific Advisory Council and all my faculty members. I should like to thank the Honorable Chief Minister of Uttar Pradesh, the Minister for Health, the Honorable Chief Secretary of Uttar Pradesh, and the Principal Secretary, Department of Medical Education, for their unstinted support to this center, and of course, giving us their guidance and how we should take it forward as an institute. Thank you very much indeed. Okay, so that I think rather than I remember all the slides, always good to take it forward, and you'll also appreciate the infrastructure that we have. So I think the goodwill ambassador of Amity, as I said, goodwill is around somewhere. And she asked me to send an abstract, which I rarely do. But I thought, let me put my thoughts together so it gives an idea to where I'm headed to. In terms of Atmanirbhar, I think when we look back even at the SDGs, you'll find that India was not only Atmanirbhar then, but it was also very sustainable the way we lived our lives. It was very, very sustainable in terms of environment, in terms of consumption, everything. I think over the centuries, because of various reasons, I think our lifestyles changed, and somehow we went the Western route and sort of lost the sustainability as well as the Atmanirbharta and the confidence, of course. So I think what happened after our independence was phenomenal. The way, even in my times, I think we were importing wheat from the US, and now the fact that we export wheat to several countries in the world. So both in terms of food grains, in terms of chemicals, pharmaceuticals, health, you name it, and we've done it. So I think another example is that of how institutions from CSR, apart from ICAR, have been able to not only double, but I think quadruple, and in some cases been able to enhance the farmer's income to almost 6 times by the use of technology that had been developed at CSR itself. So I think it's a phenomenal journey that this country has made, and we should all be very, very proud of how the scientific fraternity has been able to contribute to this journey, to where we are now. And I'm sure what the Honorable Prime Minister says and what the vision of this university is, as enshrined by Dr. Selvamurthy, is to see how each one of us can contribute towards nation building. So I think once we start thinking about the nation as the unit, then everything falls in place. If we think ourselves as the unit, then it becomes very difficult to enlarge or expand your vision to how you'll contribute to the nation. And I think that's the crux of institutions, and that should be the crux of how each one of us should think that how we are contributing to the nation. And that will drive you no ends in terms of both your innovation skills as well as how you can work towards taking the nation forward. The other thing that I feel very strongly is the frugality in our system, the Indian system. We are very frugal in nature, and I still remember as a young scientist, we were asked to develop programs which would say, and Dr. Sanjay and Bodh Chakravarti will bear me out, they would say that you have to set up a program, write a research program, which will be zero budget. So what can you do with the existing infrastructure? That you write a program first, and then we'll see. And it was very common in CSR. You had to write a zero budget program, and then we'll see what is required incrementally to take it forward. So I think it's rather than look at machines, we should look at the machine that we have, which is our brain, and see how we can utilize it to the best possible manner. Frugality being the norm. And there's so many things around you that you can utilize in innovating with a frugal mindset. So I think what epitomizes frugality and what epitomizes innovation and what epitomizes humility is none other than our former president. And also the excitement that he always had. And I still remember this picture was taken in 2014. He was the former president. He had laid down charge, and I met him in Ahmedabad while we were organizing a conference in that institute, and he came to inaugurate the conference. And he asked me, so before I go and give my lecture, you have to tell me what you do as a director. What is your area of expertise? When I told him about nanotoxicology, he sat down. He said, let's sit down and talk. The other pictures that were taken, I mean, the kind of inquisitiveness he had is remarkable. So I think that's the other thing I always take as a challenge, that I always feel I was dumb yesterday. So if you don't have the drive to learn, you don't have the drive to acquire new knowledge every day, I think then we are failing the system. So that is what I learned big time from our honorable former president, Dr. APJ Abdul Kalam. That anything you talk to him, he was always excited and what is new happening, where are we headed to? And this meeting was on genetics, and he was so thoroughly well led when he came and presented his talk about genetics and where it's headed to in the globally, not only in the country. So I think we need some role models around us whom we can aspire to become, not become, but at least aspire to acquire some of their skill sets or their attributes. The other thing I was quite intrigued, and I'm showing these slides especially for people who are maybe higher up in the ladder, but also for the people who are into the innovation paradigm. So I think Richard Miller, as we called him, he was the founder president or the founder president of this Olin College of Engineering, which is a college which does not give you a degree in a specified engineering field. And the story goes that the Frank Olin had about some $35 million, perhaps, or more, maybe 350 million. I don't remember the exact figure. And he said, I want to establish an engineering school, but I need people who can guide me. So everybody ran to him that I want to set up a school. So he said, no, I want a person who can tell me, define what engineering is. So I don't know if engineers are sitting, any engineers sitting? They are? No? Okay. So what he said was remarkable. He said, well, engineering is not about a specified field. Engineering involves everything. And this was the model they floated. So you have to look at feasibility.
We have engineers. That table is completely.
Oh, okay. I'm sorry.
Electronics, mechanical.
Very senior people, but I'll still have to show this. But I think what he mentioned was, and that's why I borrowed a slide from him, is feasibility, viability, and desirability. And that everybody knows when they start any program. But what was enshrined in these words is remarkable. When you talk of feasibility, it's engineering and sciences. When you talk of viability, it's business and economics. And when you talk of desirability, it's psychology, arts, humanity, etc. So this is what an engineer should be, according to him. And so he said, if you can design a program, a curriculum around this, only then your engineering will succeed, right? Otherwise, you will make something and you'll go out there and find a person who will sell it, but may or may not succeed, may or may not fulfill the aspirations of the masses. So the understanding of masses, the understanding of where we are headed to is equally important. So that is how he came up with this Venn diagram. And then when everything meets in right proportion, that is where innovation happens. So I think this is a remarkable way one can think, and this I'm talking about way back, I think 2012, 2013, when he gave the presentation. This is a remarkable way one can think even before starting a proposal, because generally we talk of perhaps feasibility, that what we are able to do and what is desired may be secondary, and whether it is viable or not, that is the last thing, because we always say, well, we don't know about the business aspect, so let somebody else think about it. But unless you understand everything in the right proportion, it becomes very difficult for you to innovate a new product. This I would say more for the students and the senior faculty, and pardon me for this. This is something I coined long back, sky versus sky, which is the saying goes, sky is the limit for us. That's what we are taught all the time. But the contrary of that is pratyaksham kim pramanam, which means that what you can see doesn't require any evidence. So there goes. So if sky is your limit and we are saying that what you can see does not require evidence, then where is the sky? Where is the limit? That means you have set a limit which doesn't require anything. You can just sit and relax because that's your limit. So I converted that sky into a sky which is science, knowledge, and innovation, which is SKI, just one point mutation for the people who are from biology, they will understand. And so I thought rather than sky being your limit, let science, knowledge, and innovation be your limit. And for that, of course, you have to spot the opportunity, avoid herd mentality. And I must say, except one perhaps, maybe two, all these photographs that you see have been clicked by myself. Okay, so when I say avoid herd mentality is there's a good picture of pigeons. They will always go up to a height and they'll come back and settle down where they were. None of them actually breaks that herd mentality, and so therefore they are confined to that space. Versus an eagle who will always be alone on the top, and so I think that transforms your thinking totally. Okay. Just I think 2 weeks back, I was in Mehrangarh Fort in Jodhpur, and suddenly I saw all these kites coming over or eagles coming over the fort, and I couldn't understand. Then you zoom. That's the other thing. Always have an eye for observation. Why they're coming there? There was this guy standing there and throwing food at them, and there was a big competition going on among these eagles. Which one can catch it, even snatch it, and take it away, and then they'll fly up. So I think when I say you can spot the opportunity, you have to spot the opportunity and take the decisive action after that, right? And only then that I say there's plenty of room at the top. There was a famous saying that there's plenty of room at the bottom, and that's how nanotechnology started because they wanted to look at atomic level physics. And here again, while I was assembling the slides, I'm not sure if the audience can actually see it. इसका लेजर कहाँ है? चलता कुछ करना ना चाहिए. Actually, when I was sending the sites, it's not visible here, but actually there's a drone right here. So in the larger scheme of events, can you actually see the minor details is very important when you are embarking on a mission of innovation or a mission of institution building. I think very, very important. And I thought it's a very perfect way to begin. And the other thing I always say, well, science, knowledge, innovation, people always say, well, you have to excel. I think science, knowledge, innovation, once you go into that path, you just climb a path of excellence without fail always. And the previous, rungs keep falling down. You just keep going on and on and on. And that should be your limit. Right, I think that's the way of thinking that should be in you, rather than thinking, okay, 5-day week, let's do it, and then next week we'll relax. I think unless you drown yourself in the problem you're facing or what you want to achieve, I think it's very difficult for you to achieve that in a limited time frame. As far as Atmanirbhar is concerned, as I mentioned, it started way back even before independence, and the first S&T organization to be established was CSIR. I wasn't aware that so many CSRians will be on the podium as well as off the podium, but nonetheless, it's good to know. And we only learned when we were assembling our slides for directors meeting, right? And this is the timeline in which different organizations came to the fore in this country, and they started contributing to the S&T ecosystem. And the last to come to this was the Department of Biotechnology, the youngest of them all. And also, I don't know how many of you have seen The Rocket Boys as a movie. It was only then I came to know that ISRO was actually signed off by CSIR, which is remarkable. So I think, again, when you look at how people think, I think these were visionaries who established these organizations. These are visionaries who thought of a nation that required scientific temperament, and not only scientific temperament, innovation to the core so that we could become self-reliant. And that is the fruit that we are bearing now, and we are enjoying those fruits as we speak. So it has taken time. Of course, we could have done it faster. In hindsight, we can always say that. But I think the fact that the infrastructure was built over this period of time, I think it is remarkable that the younger generation is enjoying the fruit now. So I'll just sort of take you through now a journey which I feel was very important. Just to share with you, I changed my field of research almost every 5 years. Not because I got bored, but because I thought it is getting plateaued over a period of time. So it's always good to see and ride the wave, as they say, or ride the surf, as they say. And each of these areas that I contributed to were not at that point in time existing in the country to that extent, and so therefore they were very important for the Toxicology Institute. And some of which I think I owe it to the fact that people had the appetite to sort of take that risk at that point in time. One example I'll share with you was way back in perhaps 98, when we were making the 10th five-year plan, Dr. 98, 99, I think somewhere around that time. And we were all asked to write our bits and pieces and give it to the director. So I wrote in that program that we want to start in silico toxicology. I had no clue what it was, to be honest. So when the program went to the director, he called me to his office. Do you know what it is? I said, no. Then why have you written it, sir? Unless we write in silico toxicology now, in the next 5 years, we'll be nowhere. I'm giving my guarantees that I'll go and learn what it is. I'll figure out what it is and we'll establish it in the institution. And believe you me, he said, well, Alok, I'm putting it in. I don't know whether CSR will approve or not. But if it is approved, I'm also going to put in a remark for my internal records that you are responsible for establishing it. I said, absolutely fine, sir, don't worry. When this program actually got approved, in silico toxicology was approved, we set up the facilities in collaboration with the industry. Upfront at that point in time, it was unheard of that industry was part of the program. And rest is history. Now we are among the top institutions who are doing in silico toxicology. We have a high performance computing system for toxicology. All kinds of toxicity assessments are done by computing now at the institution. So I think if you are able to visualize what is going to happen in the long run, and of course there's somebody who's able to back you or to support you for the risk, and for the leadership that's on the dais, perhaps all the dais, I'd just like to say it's a win-win situation for the leaders, always without fail. There's somebody who wanted to go to the moon. Somebody said, okay, we have a crazy guy who wants to go to the moon. Let's support him. That's all that he has to say. If he goes and succeeds, the leader gets the accolades. You see, this guy's put somebody on the moon. Nobody remembers who went to the moon, but they all remember the president who sent the person to the moon, right? If they don't succeed, the president always say, I told you, you are a crazy guy. So you died, it's your fate, I don't know. So I think both ways you win. So there's no harm in supporting the risk when somebody is willing to take it. Of course, it's a calculated risk. You have to understand what the person is heading towards, whether it is useful or not. And at times, I was just going through some of these innovation systems that we present or we get a chance to review. The ballpoint pen for students was innovated somewhere in 1888 or 1846, one of the two dates. The patent was filed then, and it got commercialized in 1946, at almost 50 years. So I think it's also a fact that people who are at the helm of affairs may or may not be able to visualize what that person is saying. But I think if you give the leeway and you monitor the person and you look at the projects and you see how they're moving in a quarterly fashion or a half-yearly fashion, depending on the quantum of the project, I think a lot of this figure out whether this is worth pursuing or not. And so the small story that I'm going to tell you now is of that risk that I took and how it was supported and where we are now. So this institution that IITR, of course, you are aware, is an institution that not only takes or undertakes basic applied translation research, but it has got all these verticals. And towards the end, we have this R&D partnerships, and something that we started was the Center of Innovation Translation Research, which was called SITAR. And I think when you give acronyms or when you sort of think of something, I think you should have a lot of thought behind what you're saying. So when you say sitar, it's actually an instrument that it's there in the Indian system, and it's very fascinating to hear or to listen to it, but as long as it is in tune or is in sync. Even one wire is not in sync, you'll hear noise, it's screechy. So I think when you talk to industry, you should have a system which is well oiled and greased and in sync and in tune with the industry. Otherwise, you'll just keep making noise both ways. You keep making noise as an academic, and industry keeps making noise as an industry, and the twin never meet. So I think with that thought, we set up this sitar facility, and we were fortunate that it was supported immediately by the BIRAC, DBT BIRAC, and we were perhaps the first one in CSI to get a BIRAC facility in the institution. Later on, it was supported by another DSIR, CRTDH, for environmental health. For those of you who can't recognize, there's still me in the center. This is 2005, a long time back. And these are all actual pictures that I click. I don't use pictures from the internet. This is the day on which this meeting happened. This is the window of my house at that point in time. Okay, so I just clicked that picture, and these two gentlemen are the professors, Professor Syed Hasham and Vladimir Tarabara, they're chemical engineers. So for the engineers, and I was a biochemist. So unless you speak to the counterparts, it's very difficult to understand what you're headed for. So this was a Friday meeting, like today, perhaps in the morning hours. And they asked me, well, Syed Hasham asked me to join that meeting just to see what they discuss every Friday. And they were discussing on fullerenes at that point in time and how they will be using those fullerenes for water purification. So Vladimir was making these fullerenes, they're both professors that time. And he said, well, Alok, if you can test the toxicity of fullerenes, that'll be fantastic for us. So that will be a step forward for us. And you have to believe me that I did not even know what nanotechnology was at that point in time. And again, something that one has to learn over time. You're an ambassador as soon as you get out of your home for your home. You get out of your institution, you're an ambassador of the institute. You get out of the country and you're an ambassador of the country. So I couldn't say that I don't know what nanotechnology is because I was representing the institution as well as the country. So I said, well, the issue is that I come from a federally funded institution, so I cannot say yes or no unless I've asked, because it's an Indo-US collaboration. So how about I get back to you early next week? Oh, yeah, that is fine, because tomorrow is Saturday, Sunday, anyway, it's a weekend. I went back home and frantically read all about nanotechnology and where Europe and US was headed to. At that point in time, I was doing primarily genetic toxicology. And then I wrote a very long email to my then director, who was in charge, who was the director of CDRI, but in charge of IITR, Dr. C.M. Gupta. This was the email I wrote, so don't bother about reading this email. This actual email that I wrote to him. But the remarkable thing is, what was the response? Dear Alok, please go ahead. Best wishes, C.M. Gupta. I think this is a great learning for leaders who are here. This is how you support risk. The next day morning, 6 a.m., I get this letter. So one thing I learned was that the leaders perhaps get up early in the morning, which I don't. That point in time. Because when I woke up, I got this email. The second was that how quickly they are able to take a decision, right? So I wrote all this about what is happening, and it will be prudent for IITR to get into toxicology. Get into toxicology of nanomaterials, and just one liner, please go ahead. I came back. Yeah, here he is. He's still very active in research. He's settled in Bangalore now. So that resulted in our first publication in 2006. So timelines are very important. As you would have seen, this was in late 2005 when I was discussing with the director. I came back, I started working, and we got the first publication out. And just for the engineers to know, this is the most highly cited paper for both these gentlemen in their entire curriculum and the entire CV. This is the highest cite. I think it's about 400 citations, around 400 citations now for this paper. For two reasons. One, that we had done toxicity of these fullerenes early on in human cells. Second was that we had done fullerenes in water rather than fullerenes in different solvents. So we had taken care that the solvent doesn't cause toxicity, and it's in fact the state of the fullerene that is causing toxicity. So I think it's very important for us to understand why there's so much buzz about nanomaterials and why there's also some apprehension regarding its toxicity. So I think forget about everything else, just look at the size. If you look at a fullerene, it's about 1 nanometer in diameter, and if you look at DNA, it's about 2.5 nanometer in diameter. So even if it is absolutely inert as a substance, it can physically go and damage the macromolecules. That was the challenge, and that's what even now the world is grappling with. And so therefore we started documenting what can be done. And a very simple example in the Indian context would be a bundi ka laddu. If you take one and then you start breaking down into independent bundis, you perhaps get exponentially high surface area, right? And for catalysis, this is a boon because then you can get extra surface area. So catalysis is very good. But inside the body, it is a bane because then if it is interacting with the macromolecules, it causes kind of toxicity for that. So I think these were issues that we are grappling with, and we brought it to the note of the world that how we are going to understand the toxicity. And also there's a paper in Science at that time that if you go down to 5 nanometer in diameter, you'll have about 60% of the molecules at the surface. So that is remarkable, and that's how people are utilizing the various facets of nanotechnology. And the other thing was, of course, how these nanomaterials, right from the raw materials all the way up to end of life, they enter the environment and they are able to contaminate the environment as well. So these were very important issues that were being addressed. So then as a toxicologist, we were looking at what all are the issues that will provide information about the product or the material that you're testing. So of course, the coating during the synthesis, the characterization, how do you characterize such a small particle? And there were several things that we were discussing at that point in time, including polydispersity, including exposure assessment. So if you look at exposure assessment, let's say of benzene, xylene, toluene in the environment, it's very easy. If you want to do radioactivity assessment, it's very easy. But when you talk of nanomaterials, how do you go to the environment and pick up nanomaterials? It was very difficult at that point in time. How do you assess exposures in the environment? So, and these, when I say that these were all documenting the things that were facing or the challenges that we were facing, it was globally that this was being felt. Perhaps we were the ones who at that point in time documented it. When you look at size and shape, so when you look at chemicals and you look at the toxicity, for example, of let's say ethanol, once you've done it, you've done it. And you know what are the drawbacks of consuming ethanol, or if you're exposed to ethanol, or benzene for that matter. But when you talk of nanomaterials, every time you change the size and shape, the entire profile changes. So how do you go back and do the toxicity every time? It was very, very difficult. And apart from all these issues, the major one was this development and validation of test models. And in fact, the fact that we had documented it later on got converted into a European Union FP7 project, which was 13 million euro project, a flagship project of the European Commission, with 19 countries participating, including myself. And this was the fact that when you put nanomaterials or you want to assess the toxicity, they themselves sometimes interfere with the test systems. So I think it was very important to understand the behavior of these nanomaterials, and that's what this project was all about. Right. So I'll just give you examples where either innovation was done, and this was primarily to undertake experiments, not to make products, or there are examples as to how an in-depth observation actually led to a discovery. Okay. So this was, of course, when we looked at zinc oxide, and of course you choose a problem which you wish to solve rather than and you wish to solve rather than looking at the equipment that you have and then choose a problem. Right. So it should be always the other way around. Choose a problem, then figure out where you want to interact and get things done. So zinc oxide we chose because it was being all the way up to CCMB in Bangalore. that didn't deter us from undertaking the study. And we did have a DLS, dynamic light scattering system. So we were able to measure that, and of course the BET was provided by the manufacturer. So as we were studying these, we found that the size is getting altered. So we had to understand why it is happening. So it turned out that each of these machines measure different parameters. So we are able to connect them. So, but as I mentioned in my earlier slides, it is important to know the size of the material. Other than that, you'll not be able to sort of say which size particles you've done. So after a lot of deliberation with the international community, it was sort of accepted that we should write all the three in our publication so that it becomes a norm. And as and when things evolve, we'll be able to figure out the exact size of the particles that were sort of assessed. Because when you measure the TEM, it is in dry state, but when you do the experiments, it's in a sort of suspension format. Okay. So when you look at morphology, this was very interesting. There was a young girl who was working for her PhD, and after a certain number of days, she came back to me. She said, "Sir, there's no toxicity of zinc oxide. Everything is fine." I said, "Very good. I mean, we don't preempt what the results are going to be." So I said, "Let's go down the microscope and see what you're doing." And this is what the results of the microscopy were. That actually, what she was doing is after everything was done, she would sort of decant the media without taking pictures, and the cells that are adhered to the bottom, she would scrape it off and do the experiments. But actually, what was happening, both in terms of the number or the amount of exposure that was happening or the dose that was given, the large number of cells were actually dying and they were thrown off. So the ones that were remaining were alive and kicking, right? But when you looked at this actually was a very clear indication. So then we actually published this plate as such in the paper to ensure that others also understand that this is equally important than just conducting the experiments and sort of reporting them. But the challenge that we were facing, especially with electron microscopy, was the fact that you can only about count maybe 50 to 100 cells, and then one has to believe the one cell that you actually published, right? So this is what it is. So when we were looking at this, and the other, for us, the pain point was we had to run to Bangalore every time. And the other thing was, of course, that's very expensive and also time consuming. And the fact that we can't use these cells afterwards because the blocks are there and you cut them and then that's it. So we thought, let's look at other things that are available. So these are some of the micro photographs which shows very clearly how the nanoparticles have actually moved inside even the nucleus. There's a nucleus and you can see the particles, and you do electron microscopy, you do the analysis of these particles, and you can figure out, right? So this was a very good observation that we came to. One of the students actually brought it to the notice that they had done electron microscopy and they found that the nanoparticles were moving from one cell to the other through the filopodia. And that was very, very important in terms of the fact that if part of the skin is exposed, maybe the other parts of the skin will also get exposed because of this traveling. And this, when we went in depth, we found out that it's similar to the transport of melanin in the skin. So as I mentioned, these constraints were always there. So what do you do? We looked at a flow cytometer that was in our laboratory, and we said, well, let's look at the basic principles of flow cytometry. And is it possible that we can use flow cytometry for looking at uptake of nanoparticles in the cell? And as I mentioned to you, everything is documented without fail. So this was the hypothesis we laid and we published it, that if when you fire the laser on the cell, the particles are sticking outside the cell, the forward and side scatter both get enhanced. If the particles are inside, only the side scatter gets enhanced. So essentially, it tells you the density has enhanced. And if there's fluorescence, you can also measure the fluorescence of the particles. So then we took titanium dioxide and zinc oxide, and we found what we had hypothesized was actually true. And we could measure the uptake of nanoparticles to the tune of about 50,000 cells per minute. And this was phenomenal in terms of the speed, and we can actually take these cells and sort them out as well and use them again. Okay. Then we looked at progressively, once we know that these particles are getting inside the cell, do they interact with the DNA as I showed you earlier? So these are some of the things that we do in the laboratory. One is the damage to the DNA, which is repairable. Then you go to, if it is not repairable, the cell will die. But then if fragments of the chromosome are broken, you can again do chromosomal analysis or micronucleus assay. But if this goes on and is expressed as a mutation, you can do the mutation assay, and then if it grows, it becomes a tumor. So all these things we were able to figure out. And I must mention here that we improvised these experiments every time we did, because we didn't have any positive control nanoparticles then. So we were using ethyl methane sulfonate, which is a known carcinogen and damages the DNA. And what we found that using very, again, the concentration that we were using was what is left over in your hands after you wash off your sunscreen, right? So those were the particles or the amount of concentration that we were using, right? So again, with the micronucleus assay, and this was very interesting again, that we published, was the fact that if you are using nanoparticles, especially titanium dioxide, they go and stick to the cells even after they are washed off, and they are actually interfering with the counting of micronuclei. So they look like micronuclei themselves, and sometimes they interfere with the counting. So we published this plate as such again, for others to sort of take care. And one thing that's not here, we developed a micronucleus counting method using flow cytometry as well. So that becomes a 3D method rather than a 2D method, which is currently available. For these nanoparticles, we actually then came with a hypothesis. Again, these are all published data that I'm showing you, that once these particles enter the cell, it's all been proven by electron microscopy. They actually go inside the nucleus, they damage the DNA. They also damage the membrane through lipid peroxidation, reactive oxygen species are formed. The mitochondria get influenced and they release pro-apoptotic factors, and ultimately there's cell death. And this, again, something I must say that depends on what you do. In January of 2009, we published this work. I think February or March, we were approached by the European Commission that we'd like to publish an interview by you. So I said, that's not possible because I'm a government institution. So if you send me the questions, we'll send you the answers after approvals. And within 3 months, I think April, they came up with a special issue for their policy. And they dedicated one full page on the kind of work we've done. And they said that this is the kind of data that we require to provide so that we can take appropriate decisions regarding the regulatory framework. And currently it has more than 720 citations on this publication. Most of our electron microscopy work has been published after taking due recognition, copyright transfers by Elizabeth in her book Sunscreen and Biohazards. And we also looked at the environment and the whole gamut of species that are in different trophic levels of the environment and wanted to see. So I'll just give you a snapshot of what we were able to do, especially in the bacteria. Again, there was a challenge as to how do you measure or monitor uptake of nanoparticles in bacteria. And with our experience with human cells, we thought, let's try flow cytometry again. So we first looked at the internalization of zinc oxide in E. coli, and we were very glad to see that we could monitor the side scatter as the amount of particles were getting increased. Same was true with titanium dioxide. But this was something path-breaking, that while we were doing our electron microscopy to validate our results, the student said that here is a E. coli that is dividing and the nanoparticles are still inside, which means that if the nanoparticle or the bacteria is getting exposed in the first generation, even the second generation is still having these nanoparticles inside them. And so therefore we looked at a study that was a multi-generational study, which was 3 generations. And even at the 3rd generation, we found that nanoparticles are still existing, both titanium and zinc oxide. And this was concurrently validated by electron microscopy all the time. Yeah, these are some electron micrographs that you will see. for Salmonella typhimurium in Ames test. And of course, we kept um, looking at more and more data, corroborative data. So here was a scanning electron microscope with elemental detection to look at whether our nanoparticles, when they are inside the cell or when they are coated with proteins. So you can see zinc and oxygen to begin with, but when the protein is coated, you'll find nitrogen, carbon, oxygen, and zinc all are available. So it was all to prove beyond doubt that what we are saying is correct. And here was the outcome, that the student got a novel method for detecting uptake of nanoparticles in bacteria. This is the first time somebody had reported using flow cytometry, published in the journal called Cytometry A, which publishes only new methodologies in the area of flow cytometry. Another thing I must mention, especially for the students, you should come with an open mind when you start your PhD program. There's a very good example that I always show. Lokesh Baveja was a gold medalist from the JNU biotechnology program and he was from MS University Baroda. Came to do his PhD with me, and the first day he said, "Sir, I want to do mole bio. That's it." I said, "Well, we'll decide about your program. Don't worry." At that point in time, another PhD student was going to Pune to learn how we can do this kind of modeling studies and nanoparticle-based in silico approaches with the industry. So I said, why don't you go with him, spend 2 months in Pune, learn all about this, and then we'll look at, in the meantime, what we can do. So he went and came back, and rest is history. He never, ever touched a test tube. Right, and this is a brilliant example, and he's got several international publications with international partners in collaboration, because they were doing biochemistry and he was doing all the simulation part. So I think this was a brilliant thing to do. So he looked at graphene oxide, and these red dots that you see actually, yeah, are the hydrated parts, and the other one, if you look at the protein, which is an alpha helical protein, where there's no hydration over a period of time, it loses the helicity. Right, so basically, this is how interdisciplinary you can get. It was a 100% biology problem, solved 100% by computers, and published in a 100% chemistry journal, which is Langmuir. Okay, so why we did that was the fact that at that time, graphene was in major demand because they wanted to use it as a sort of drug delivery agent, and they wanted to see how graphene can be utilized in for drug discovery and drug delivery processes. So I thought this would be a good journal to sort of send it to. And over a period of time, as Dr. Selvamurthy mentioned, I was able to set up the entire repertoire of right from synthesis all the way up to in silico prediction, everything that was required for engineered nanomaterials. ENM stands for engineered nanomaterials at the institute, which is CSIR IITR. Of course, we conducted a network program in which 17 institutes participated, and IITR was coordinating this program. So other institutions were basically synthesizing materials, and we were testing the toxicity and whether they were suitable for the application or not. Again, documentation, I just put these last night. I thought it was very useful, especially when going to a university like Amity. This was the first international conference we did, I think 2008. So there are only two ways to learn. Either you go to every institute to find out what is happening, or you call the people and learn from them firsthand, okay, including myself. So what we did, I proposed that let's start an international conference. We started nanomaterials in 2006. This was 2008. Let's do an international conference. Bizarre idea at that point in time. Director said, नहीं कहाँ आप कुछ करते नहीं, हम लोग we just started. मैंने कहा सर हम पूरे वर्ल्ड में घूम के सीखें, इससे अच्छा सबको यहाँ बुला लीजिए. Let's learn first hand from them. And believe you me, anybody we invited, they came, including the lady who wrote the National Nanotechnology Initiative of the US. She came and inaugurated. We had the best of people from the globe who came and presented. But then we thought, let's document it. So we wrote down, and that's where perhaps Chakravarti, I don't know whether you were there in CSIR or not, but nonetheless, 2008? You were there. We wrote, I think, 6 or 7 pages of what we did, and more than that, what are the recommendations? That is very, very important. What do you recommend? If you call such people, you should take the maximum out of it. What do you recommend that the organization or the country should do? So each one of them, the foreign countries that come, they provided they provided their recommendations because they were stalwarts in the field in their respective countries. We documented all that. We sent it to the director of the institute, and I was responsible for this. And when we sent it to him, अरे institute का letterhead में न्यूज़लेटर में तो आप 8 पेज अपना डाल देंगे तो बाकी का मैं क्या डालूंगा, आप इसको काट के 2 पेज कर दीजिए. इसलिए मैंने पूछा चक्रवर्ती साहब को कि सीएसआर में थे कि नहीं, तो वो हमारे न्यूज़लेटर में 2 पेज का छपा है. सीएसआर को मैंने जब भेजा तो वहां से फोन आया, सर ये तो बहुत अच्छा डॉक्यूमेंटेशन, मैं पूरा छाप दूं क्या? मैंने कहा आप छाप दीजिए. तो वर्बैटिम जो हमने भेजा था छाप दिया 8 पेज उन्होंने. फायदा मुझको क्या हुआ? दिस न्यूज़लेटर इज sort of, it's got an ISSN number, so it is always on the CSR website. Anybody can pull it down and see what were the recommendations made by this body, August body, and how we've implemented them over a period of time in this country. So this was the basic foundation of nanotoxicology in this country and how we progressed. So I think it's very important, Dr. Selvamurthy, that when we organize such international events, let us tell the government, or in our case, our headquarters, and that then transformed into telling the government as to what we expect going forward and why we have taken such decisions. So I think it's a fantastic way of documenting things. And based on that, of course, we got 2 UK projects. Again, the only one who got back-to-back UK 2 projects, 2 European Union projects. And we then wrote, so if you have created some knowledge, might as well document it. So we wrote guidelines. And again, we did a conference in 2011. 15 countries participated. The then DG of ICMR, Dr. Katoj, inaugurated. And without fail, you'll see Dr. CM Gupta standing here. And we released a journal in which all the abstracts were published, and alongside that was published the guidance document or the guidelines for safe handling of nanomaterials. And this became the basis for the subsequent documentation of policy of the government of India. So again, what you think may or may not be correct at that time, I don't say it was 100% correct, but whatever knowledge that existed at that point in time was documented in the form of guidelines, and it was presented to the world. And as we had or generated more new knowledge, the policies came forward, and we were also part of the policy frameworks of this country. This I've already shared with you. This was another remarkable thing that happened, that we were able to train 8 countries in the area of nanotechnology or nanotoxicology, and they went back and established nanotoxicology in their respective countries. Again, that was remarkable. It was a turnaround from what we were doing. We go abroad and learn and come back and establish it in the country. So we organized a workshop. Again, I think if you have knowledge in a certain area, do train as many people as you can. Now, this I'll just like to show before I close the timelines and the speed and the scale at which it was done. So as I showed you, the email was in November 2005. I came back to India in February of 2006. April, we started setting up the facilities. December, the first proposal was submitted to Department of Science and Technology, which got funded. Established the Indian Nanoscience Society in 2007. International conference organized in 2008. We got 2 UKRI projects between 2007 and 12. Another 2 European projects in between 10 to 12, and 1 from 12 to 15. And CSR took notice of this, and they gave us a network program called Nano-SHE, Safety Evaluation of Health and the Environment, between 12 to 17. We published the guidance document in 2011 with 15 countries participating in the next conference. We set up the Advanced Imaging Facility, which was established in 2016 by the Honorable Minister Dr. Harshvardhan. CSR was globally positioned among top 5 in nanomaterial toxicology by 2017. We were number 4, and number 5 was Harvard. 2017 July, I was given the honorary causa DSc, like Dr. Selvamurthy mentioned, and I wouldn't have put this up. Doesn't behove me, but I think why I put it up is the following. In the history of that university, which was about 52, 53 years old, I was the first person to be given an honorary doctorate outside of the UK. Right, so I think even in their scheme of events, it fitted to give an Indian who was associated with them for long and has contributed to the field from India. I never went abroad and did any research in nanomaterial toxicology at all. So I think if you really have the passion to do something, India has enough to offer. And then we published our first book on nanotoxicology in 2017, and subsequently CSR came up with the National Nanotoxicology Facility at IITR. One of the first guidelines were published by the Department of Biotechnology and ICMR in 2019, of which I was a part. Again, the second guideline came out in 2020 on agri inputs and food products. Again, I was part of these guidelines. These were the first one released by the Honorable Minister. And these are freely downloadable from their website. Subsequently, the European Commission in the Scientific Committee on Consumer Safety, they have cited several of our papers in their guidance documents of safety assessment of nanomaterials. The Nano Mission of India, they came up with the guidelines. Again, they quoted our guidelines as well. Published 3 books, one after the other, on various aspects of nanotoxicology. It's all documentation. I think it's very important. Of course, the backbone, very important. All of them are either abroad or faculty somewhere or the other. I think without them, I wouldn't be standing here. They've contributed tremendously, and they were able to bear with me. I think that's even more important. Now, when I say supporting risk in science and a focused approach helps achieve the goal, I think you would understand what I mean by this. Again, a photograph taken by me, I think the day after Christmas in Michigan. That's only later on I realized there's a lot of science in this photograph. So when the first bird flies, it creates a vacuum for the other two, and then so on and so forth. And when the first bird gets tired, the second bird takes over. So when you look at these formations of these geese flying in the sky, you'll find a L to V formation over a period of time. So that's how they fly. But the more important lessons for leadership was that, once you've done your bit, back off, let the other one take forward the mission or goal of this, the organization or the program that you started. So I think it's very important. And for them, it's important to take that risk because they are flying from one country to the other. If they don't trust each other, they will fall dead. So I think everything is at stake when you talk of science, when you talk of institution building. So trust is a very, very important inherent part of institution building. In terms of science, this was again a photograph that I've taken from IIT Delhi alumni website long back, but I thought it was very intriguing. They didn't write anything on it. They just wrote alumni of Delhi IIT. But I thought it has a lot of message in it. One, when you talk of EMI, we all bothered by EMIs for our house, for our education, so on and so forth. But for science, the EMI is earnest in observation, which you've seen. 100% motivation, 100% innovation. And the thing I always tell the students, something somewhere terribly went wrong, was the fact that we started with this gate. This was Stone Age, this was Iron Age, and now we are in computer age. We are back to where we started. We don't apply our minds at all. So please, for God's sake, forget about Google. Apply your mind. There's enough in your head that you can take things forward and innovate. At the same time, while you're innovating, you should be very careful and mindful of the environment we live in. We are just one species in millions and millions of species on this planet. We already made several of them extinct. There's a beautiful photo of a human face, which is there because of all the animals that you see in that picture. So as you make these animals extinct, human will be extinct within no time. So I think we should be very careful in how we innovate in our systems. I always say in science, if you don't have the eye for observation, rather than science, I think anywhere, if you don't have the eye for observation, it is meaningless to do that thing. So I think here, the same set of experiments or same set of photographs. It depends on how in-depth your observation is. So here is a person you can see at this sort of level, or you can further go down and see the person inside the eye who clicked the photograph as well. So it depends on the depth of observation that you make in the experiment that will decide the inference of what you've done. Right. So the more inquisitive you are, the more you'll derive from the same experiment. Again, this was a photograph taken in Calcutta, and it's about 6:30 in the morning. One of those days I got early, perhaps, but that was primarily because we had to reach the conference site for breakfast at 8. I was very intrigued. In those days, we had a very small camera with 2-inch of, I think, LED at behind it, and you had to download the photograph to see it. So I immediately downloaded it on my laptop to see what this bird was, and very intrigued by the kind of bird. Ultimately, it was actually a crow. This was the feather. This was a ray of light coming through. Now, unless you have the, I'll keep saying it, you have to be inquisitive, you have to be curious, you have to have a very strong sense of observation. Only then you'll be able to succeed. This is something which I always say from WhatsApp University, something useful I got, which is life is always about how we see things, for sure. And I am very grateful that the Honorable Prime Minister has added Jai Anusandhan to the earlier Jai Jawan, Jai Kisan, Jai Vigyan, but not undermining the fact that right from Jai Jawan to where we are now, everywhere it's the science that has prospered. It is the science that has allowed those Jawans to be at the borders and defend this country. So I think it's a huge honor that we as scientists represent this country. We are able to contribute in whatever little fashion we can to this nation. And the title that you see, not Atmanirbhar Bharat, but supporting risk in science, evolution of nanotoxicology in this country, was the title of the inaugural of TEDx Lucknow. I was invited to give the inaugural talk for this TEDx Lucknow, and I think I have been very fortunate for several reasons. Of course, my dad was a scientist, so he inculcated a sense of inquisitiveness in me, allowed me to question everything at home, and he would answer. My mom would get flustered, no, kya hai sawal poochh do? But he said, no, let him ask. So I think it's the ecosystem you are groomed in that allows you to take these risks and move forward. And I was fortunate that I had a lot of leaders around me who sort of allowed me to take that risk. So with this, I'd like to thank you and for giving me this time, and of course, Dr. Sanjay and Dr. Selvamurthy for giving me this opportunity to share not only my thoughts on how we foresee the country in terms of Amritkal, but also at micro level, how individuals and institutions can contribute to the nation building process that we are in. Thank you very much indeed. Thank you.
Let's give a big clap for this wonderful, fascinating lecture. Great lecture, Anu. I have heard you earlier, but then this was one of the fantastic lecture. Come, please come. See, the way he articulated the journey of his achievements, accomplishments in research, innovation, in the domain of science, technology, in many institutions which he has led and which he has founded. It's a great journey, and wish you all the best for many, many more decades of contributions to nation building through science, technology, and innovation. And also your research in nanomaterials, safety. It has really opened as an eye-opener to the country, to the world, that when we are looking at exciting things happening using nanomaterials, we should also be concerned and also be cautious. In any technology, you need to look at the other side of the coin, that safety. You brought it so well, and then you are one of the pioneers at the global level to bring this awareness and doing experiments, not just empirical, but here you have really done documentary evidences, and I'm sure this will be making a history, and you will be remembered for this initiative in India for the nano safety, nanomaterial safety. Now, I think there are many questions and maybe some points of discussions which our people would have. Yes, we'll start with Dr. Nutal Kaushik. You know her. She is the Director General for Amity Food and Agricultural Foundation. Dr. Nutal Kaushik.
Thank you. Very good presentation as always. But I have just one simple query. Like you have been like, suppose I have a dust sample, okay, and if I visualize in the electron microscope, how do you identify the? How do you give the chemical identity to different toxicants? Mhm.
So in most of the microscopes, there's an attachment, electron microscopes or scanning electron microscopes, both of them, which allows you to do elemental detection. I showed you one of the pictures. It's generally they call EDAX, where you are able to undertake the chemical analysis of whatever particles or cells that you're looking at. And it's got a library behind it, so it immediately tells you what are the chemicals, and you can take the spectra, and that spectra can be taken by.
Okay, thanks.
Just wanted to make an announcement to Dr. Alok and colleagues. Founder President has been attending the lecture right from the beginning, and he has sent a message. This is one of the best lecture he has heard, and also in the first half of this 2023, this is the fantastic lecture. So let's give a big clap to Founder, that in spite of all the busy schedule, he finds time to listen to such lectures and also implement them, not just only listen. Now he will be looking at from every one of you. What is that you are going to do out of the lessons learned in this lecture for a sustainable outcome? Our group captain Kapil Shukla is sitting there as director outcome. That how would we implement some of the lessons learned from this wonderful lecture? So founder president is actually yes.
I just have a point to make. हम हिंदी में बोलेंगे हमारे ख्याल से आग लगाना मेरा काम था, अब डाउजिंग आप लोग करिएगा. मैंने आग लगा दी. I think that is proven now. बाकी आप लोग समझिए. Yeah, please go ahead.
Yeah,
sorry,
sir. Nice talk. I learned a lot. So my question is about the zinc oxide, because we have published a couple of papers on the nanoparticle, and while we were working on the zinc oxide nanoparticle in Hyderabad, I found that these nanoparticles go and stick in the kidney and disturb the ventral pore and disturb the whole blood filtration, part. So from that point of view, and you also admit that they are being used for the cosmetics in some or other way. So what is the way? Is there any way we can minimize the toxicity of the zinc oxide nanoparticle? Number one, and how we can make it more bioavailable, so that it can be used not only for the cosmetic but also an injectable. Can we think of some strategy for converting this into the injectable, so that we can frequently use it load for the drug delivery or something?
So generally, let me answer your second question first. Generally, you do not use metals or metal oxide for drug delivery. Generally, you use polymers that are biodegradable. Because even when they were looking at carbon nanoparticles, they figured out that after one injection, they were basically looking at radioactive carbon, and they found out that just one in vivo injection in the mouse, 98% of these particles were out, 2% remained in the body, right? So if you are going to look at drug delivery for zinc oxide or any of these materials which are going to stay back, I think it's not a good idea, because then if the drugs are going to be used for chronic or long term, becomes very difficult for the person to handle, as you rightly mentioned. So that answers the second question. When you put zinc oxide into the system, I think one should be mindful as to why we have done it. I don't know why you put zinc oxide in vivo so that goes and sticks to the kidney. I don't know. And that is why I mentioned that when we look at the model, it should be commensurate to the usage of that molecule. Titanium dioxide, I agree, because titanium dioxide is used even in medicines for whitening. They are trying to see what they can use other than that. But primarily for drug delivery, they are trying to use PEG or other such materials, PG and other materials, which can go inside and biodegrade themselves for targeted drug delivery, as you rightly mentioned, they are trying to look at how in PEG itself they can embed certain antibodies or something that will identify the target cells only, and then they will take it forward.
No, actually, I asked the question because zinc is required for the matrix metal proteases and the integrity of the barrier integrity. So that's why
So I'll come to you. So the second issue is that, do you want to really nanotize everything and anything? Not required. So that's part of the guidelines now. Now it's part of guidelines. If paracetamol is fine, why do you want to make nano paracetamol? Not required. Okay. So if zinc is fine, you don't have to nanotize and send it in. Not required. Now, if you look at metals, because I did say you generally don't use metals and metal oxides. Some experiments that were done in vivo were that with gold nanoparticles, especially with the triangular gold nanoparticles that were used, and they were able to take it all the way to the affected part where cancer was there, and then radiate it from outside. And once the sort of gold nanoparticles absorb that radiation, and the radiation was switched off, they release the heat inside, and the cells, only the cancer cells died. So I think that makes a lot of sense. And the government allows you, if you are going to use nanoparticle therapy for diseases such as cancer or HIV, where even if you can increase the quality of life, let alone lifespan, even if you can improve the quality of life, they will permit the drug. No questions asked. Even if it is toxic, no questions asked. Right? Because you're improving the quality of life. You can improve the quality of life, could be in terms of pain, in terms of, increasing the hunger so that person likes the food. In terms of improving lifespan, it is a win-win, because then, so there are a lot of drugs that came early on with nanomaterials that were allowed because of these conditions. And even now they're saying that because a lot of pharmaceutical industries wanted to invest and they sort of pulled back a bit because they didn't know how it's going to pan out. So they're saying that as long as you can prove the risk versus benefit, we are fine with it. Right. So that is also enshrined in the guidelines now, that as long as the risk is addressed and the benefit outweighs the risk, we are fine. You can take it forward.
See, technology is always a double-edged weapon. Atom you can use to generate energy or a nuclear medicine. You can also use it to make a bomb. So nanomaterials, tremendous positive beneficial effects are there. Nanomaterials, whether it is nano agriculture or nanomedicine, any area, it is tremendous. But then keep it in mind the safety aspects. That's all the point which he has raised. Not that you shouldn't use it, but when you are using nanomaterials, think of the health of the individuals or the biosphere as well as the environment.
The other thing, sir, I'd like to add what you just mentioned very rightly, is that these nanoparticles, because of their sheer size, can actually cross blood-brain barrier and blood-testicular barrier. That is also, we've done some studies we didn't show here. So I think that again has come into the guidelines, that if there are any particles that have the tendency to cross the blood-brain barrier, even for the drugs that are in market, you'll have to do studies to prove that they do not cross the blood-brain barrier, that they don't cross the blood-testicular barrier. So I think these are things that are evolving as the science evolves. So we'll take it up. Yes,
thank you. Yeah, Alok sir, I know your work from beginning.
Just introduce yourself.
Oh, I'm Professor Biswas. I'm a director at Amity Institute of Molecular Medicine and Stem Cell Research. And you have not shown these slides because in the IIT,
he has published papers in hepatology, and liver specialist and liver cancer.
Even I'm happy to see your slides also in HepG2 you are using for the toxicology this zinc oxide study. And rightly you have said that is you should go for the polymer which is biodegradable for the targeted therapy. That kind of the work we are doing in Amity, Shrimati sir is telling that, because in the HepG2 one receptor is called acylglycoprotein. So it's overexpressed. So we make one, it's ligand called lactobionic acid, and we make one dipeptide on it, and then dipeptide tag with the lactobionic acid, then given for the targeted therapy and the preclinical study we did here. So it's nice your talk because I know, because in the what I am mentioning is that because in the 2018 there is a IITR one conference was there, that is a apoptosis and cell death, and Dr. Subhash is my friend, so I was invited to talk, and probably I have seen your laboratory also. So it's nice, and my I have a more question rather than I want to say that is how we can start with your place and in our institute, because we have a flow cytometry facility and all, and we are working on the targeted drug delivery system and the cancer biology and all kind of the cancer specialist in our department. So yeah, toxicology also one important part. So we can collaborate. Yes.
Yes, I think two things I'll mention here. One, as far as IITR is concerned, they have a very strong nanomaterial toxicology group.
Yes.
So you could perhaps contact either the director there and try to see what kind of work you can do in collaboration with them.
Definitely.
As far as our institute, the current one that I'm heading, the Center of Biomedical Research,
Biomedical science. Yes.
concerned, what we are very strong at is metabolomics. Right, so
great.
that is one thing we can definitely address as far as nanoparticle science is concerned. And there's another guy who works on liver related, I think, GPCRs and other things that he does, Vishnath Maiti. You might want to look at his, he's just published PNAs this year.
Okay, great.
So I think it might be worth to have a go with him. He looks at, I mean, basically we look at more of samples, patient samples, and how we can benefit the patient directly. But of course, a lot of fundamental science goes on. But for metabolomics, you're more than welcome.
Metabolomics, all setup you have there in the new.
So
center,
metabolomics, we have LCMS, QTOF, as well as NMR. So we feel NMR metabolomics, because we do it for patient samples, we are much more stronger in that field. So we can do it both in body fluids as well as in bone and in cartilage directly.
Okay.
So we have a solid state NMR which can take care of bone and cartilage directly. Direct samples can be measured, and a 800 megahertz solution states NMR, which can take care of body fluids. So both can be done this.
Okay, fine. So I'll contact. Yeah,
sure, most welcome.
Because that the toxicology, that proteomics setup we need actually more.
Sure.
Thank you. Can we give the mic to Dr. Seema Bhatnagar, then Dr. Kamal Raval?
So,
She is our synthetic chemistry specialist. She has done in silico modeling of drugs, and she is actually making a triple conjugate. Why don't you mention? Go ahead.
So first of all, good afternoon, sir. I'm also an alumni of CDRI, so I'm very happy to see you today here at Amity. The reason, what I want to talk about is, we had written to your institution, Dr. Neeraj Kumar. We had submitted a metabolomics project, and of course, some more work is required for that. So we'd like to revive that with your institution and move ahead, because
Neeraj Sinha, you mean, right?
I am Neeraj Sinha, sorry. So we did take consent from the institution, but little bit more we want to revise it and work ahead on that.
So I just mentioned to Dr. Selvamurthy is that even for Amity, we could look at it. So all the infrastructure that we now have, because it was a government institution, some approvals were required, but we got it through. And so all the equipment that is on campus is open for anybody to use at a user charge. But for research, we can always have a joint project. That's a separate issue. But anybody from industry or academia, they want to come on and just say, analyze my sample. You can do it on a payment basis that's already available, and there's no issue. And he also has the brochure for that. So
yes, sir. So at that time, that was getting decided.
Sure.
So maybe I will write back to you.
Sure.
Why don't you mention about your triple conjugate just briefly?
So, sir, we have designed a triple conjugate where we have used anti-cancer drug curcumin, and it's a PEG conjugate. We've also used some self-immolative type of linkers where we have conjugated the drug with folic acid, anti-cancer drug, and curcumin. And these conjugates are working very well in the in vitro system. Now moving to the in vivo system. So
you बैठे आपके सामने in vivo बैठे.
No, we collaborate with Dr. B.C. Das, who's the chairman.
It's a very novel approach, Alok, that we are looking at a cancer stem cell, not only cancer cell. And it's very difficult to make it, conjugate where you synthetic molecule with the natural molecule. So she has done that.
Yeah. That's fantastic.
Great.
Fantastic.
Anybody else? Yes, Kamal. Kamal Raval is our bioinformatics. He has developed a AI platform for vaccine development and the drug discovery.
Wonderful. Thank you for a wonderful talk. And my question is very generic. What
Don't ask me about bioinformatics.
No, I'm not going to. I'm going to ask something more.
Please, please.
What steps are you taking in the advent of artificial intelligence and the other disruptive technologies to future-proof your research, or augment your research capabilities in the new
Fantastic.
institute? Thank you.
I can go. Okay.
Yeah, please.
So I think very pertinent. Please have a seat. A very pertinent question that you said, and we've already been collaborating both with companies and with IIT Kanpur on looking at AI as a tool to be able to predict various disease conditions based on the already existing data that we have. So currently we have about 25,000 patient sample data for metabolomics from about 50 disease conditions. That is one set of data. The other set of data is about, I think, 8 or 9 neurological disorders for which we have functional MRI data. And what we're trying to do, and I shared 2 examples in the morning with Dr. Selvamurthy. In one case, we were able to identify using the neural networks and the plasticity of the brain. We were able to identify whether an intracochlear implant would be successful or not. So prior to operation, the PGI doctors would send the patient to us to assess whether the brain plasticity is good enough to adopt the implant that they're going to put in. And I think in a year's time, they sent us about 21, 22 patients, out of which we told them that 18 of them will succeed, and it was a 100% success rate. So that is one. So we're trying to see how we can utilize these models, and we are trying to upgrade it. So I think January, February this year itself, in one of the projects that we wrote for Department of Science Technology, they've given us a high performance computing system only for MRI related data analysis and how to build models on that. The second one was on attention deficit hyperactivity disorder, ADHD, where the child goes to the clinician with his parents at about the age of 7 or 8, and they're able to assess whether the patient is having ADHD or not. And the time frame is only up to the age of 12. So if the patient is not able to recover, then the patient remains like that forever. But based on our researches, we were able to show that even at the early age of 3, we are able to tell the parent whether this child is going to become ADHD or not. Over a period of time, we've done so many samples with a CT 3 Tesla functional MRI machine. So again, the doctors are now using that system, but we are evolving the models. We are also in touch with Siemens. So this is again something that we can do. If you evolve something really great, please talk back to the industry, means the people who develop that software. So we are talking to Siemens. Can they include our model in their software? It becomes part of the software globally. Same is true with Bruker in NMR. We are discussing with them the different algorithms that we designed to assess or analyze the data. Can it be included as part of the clinical metabolomic software of Bruker? So I think it's very good that you raise this point, and AI is definitely disrupting the field. There are a lot of companies like Roche. They've already come forward with AI integrated platforms for pathology. Some pathologists in Lucknow, I know that they're already using those for primary sort of reporting of the pathologies, and then they look at it and they are able to tweak it a bit, and that becomes memory for the software again. The next time it will correct. So I think it's a beautiful thing that you mentioned, and we should take it forward.
So we are also working with oncologists we have developed a non-invasive low-cost system where a lung cancer can be diagnosed using histopathology as well as PET CT scan, and we integrate with the genomics data.
Sure.
Now we are launching a scale-up operation. We are going across India with different hospitals. That is and we would be happy to collaborate if we can, bring in our expertise and, jointly do something, because we are very strong in imaging, deep learning and other things.
Sure. So I think we just hired a professor, Sanjeev Singh. for data analysis, and he's been there for a long time doing this kind of work. So I'll definitely connect you with him. No issues.
Thank you.
You were recently in Baylor College of Medicine.
Yes.
Why don't you mention
Yeah,
about the
last
collaboration?
month I visited US, went to Boston, met, delivered a talk and, met over 40, 50 executives in BioIT. We had a, great discussion. So I introduced this, technology over there. Then I went to Baylor College of Medicine, where we have been collaborating for past 4 years. And our AI-based vaccine development platform is being, again scaled up. Baylor has already brought in a lot of vaccines into the market. One was Corbevox, if you have heard about that. That is manufactured by the Indian Biological E company. The 100 million doses of COVID vaccines have been. It's called open source vaccine. So we are not. The Baylor is not patenting. Similarly, we are also helping scientists across the world so that our AI technology can bring down the cost of development of new vaccines by 30-40% or so.
So the only thing I would suggest, I mean, you can definitely develop AI models, etc. You can still copyright them to you and make it freely available. Nobody's stopping you.
Yeah,
it remains in your name all the time.
Yes,
that's my only limited point.
Thank you. Now, first Tanu Jindal, then we'll go to Sandeep Arora. Can you send the mic here, Tanuji? We have a formal MOU with CSIR, and he is willing to help. You can use any equipment if it's a collaborative mode, partnership mode, or on a payment mode from your project. So if you have a project where you can cater for that, all that, here Dr. Alok Dhawan is willing to help you. So please make use of it. Yes, Dr. Tanuji.
Yeah, first of all, thank you so much for such wonderful talk. And you have been earlier with the STOX conference, which we organized. You were one of the chief guests in that. And I would like you spoke about in silico toxicology. So we have published a book on organophosphate toxicity with the Springer. And in that, we have found a novel antidote site with the help of in silico toxicology for organophosphorus poisoning. So with this, new frontiers of toxicology are also have been developed. For example, polar ecotoxicology, then electromagnetic radiation toxicology. So I would like to know if your institute is also working in the similar directions.
So the current institute, which I'm heading, Center of Biomedical Research, we don't do toxicology at all. Yeah. As I mentioned, we are more into patient care and how we can improve patient care. And for that, if we need to do any biomarker identification, validation, etc., that is what we are expert in. But if you want to do any even toxicity studies or toxicology studies related to metabolomics or genomics, I think we can definitely help you in that. But that's not our aim as an institution. So even the chemistry that was happening earlier was very fundamental chemistry, and that was very evident that we are number 4 in this country as far as chemistry is concerned. And this year itself, we published in Chemical Society Reviews, one of our faculty members, a review on borylation chemistry, and that impact factor of that journal is 63. So that only shows that they're doing fundamental chemistry, they're still doing, but now what I've asked them to undertake is chemistry of APIs, for example, that are required for the country. So a very, what should I say, simple way to motivate them and yet focus them on issues would be that any money that comes, that's what I've done, so I'm just sharing the experience. Any money that comes from the institution, has to be utilized in problems identified either by the institute or for the mandate of the institute or for national missions. Any blue sky research you want to do, research, not innovation, research you want to do, you get your funding from, different funding agencies. So just to give you a sense, we are a very small institution. We are only 12 scientists as of now, only 12, about 67 or 68 research fellows. This year we published about 71 articles with an average impact of 5.73, which I think is phenomenal. We got some 6 patents this year. So I think this way we were able to even harness the expertise of chemists towards drug development now. So now they're collaborating with CDRI. CDRI has given us a target. Target means what they want to target, right? That target, not the target otherwise. So in that collaboration, CDRI provides the target. Our in silico guy is now looking at molecules that will be good for that target. The chemistry guys will synthesize them, and that synthesized compound will then be given to CDRI for their in vitro and in vivo efficacy work. So I think that's how collaboration should be devised. That will be win-win for both. Please.
Thank you. Now just wait. Last remark from Sandeep Arora, the Director of Amity Institute of Pharmacy. After that, I would request Honorable Founder President. He is going to make concluding remarks and also give your blessings for all these participants, and also to our Dr. Alok Dhawan, who is now the great leader in this. Founder President will also give some blessings to all of us. Now, Sandeep Arora.
Dr. Sandeep Arora, Director of Pharmacy. So just wanted to have your take on the recent trend, the pharma industry, as it is moving from the synthetic to more and more biological molecules. So the more appropriate route that is being concluded upon is possibility of inhalable routes and delivery systems. And micro and nanoparticles are also being regularly worked upon. You have done a lot of work on these things. Your take on this?
So my take on this is the following, and you'll have to pardon me because I have limited knowledge in the field that you excel. But the little bit of interaction that I have with pharmacists, what I gather from them, that for inhalable solutions, nanoparticles are not the good choice because they bounce back in the lung. They don't go and settle down. So they perhaps want microparticles, which could be in the range of about 2 to 2.5 microns. That will be helpful. The other thing is that they're saying rather than take any, let's say, even PEG or any other carrying agent, drug delivery agent, you can actually, so this was a lot of research going on in an institution called Institute of Pharmaceutical Innovation, IPI in Bradford. They came up with the first patent on inhalable insulin, right? So what they are doing is that they are breaking down the APIs into their smaller parts and using them directly rather than taking any carrier at all. So they're saying that reduces because everything else is worked out. So the other thing that I have heard, or I would say in discussion that has come up, is that when you look at encapsulation, the efficiency of encapsulation is not very good, right? So you have a lot of empties that go into the inhalation process. I think it'll be good to look at currently if we can utilize. In case of biologics, you might need a carrier to take it forward inside. But again, we'll have to look at things that are more biodegradable. But for APIs, we can use them directly. That's what CDRI is doing right now.
Thank you, Dr. Alok. Now, may I now request Honorable Founder President, who has been listening to the lecture all through the presentation right from the beginning, and he has also been giving guidelines to who should be the people who should be invited, and they were all chosen. Each one of you was chosen. And thank you, Founder President, for this great honor for us, prestige to have your participation. Can we have your blessings to all of us? Over to you.
Dr. Silvamurthy, there is a German proverb, your desire is an order for me. If you desire that I should speak, then it's an order for me. First of all, I must appreciate my brilliant scientists, HODs, HOIs, technocrats sitting over here. I told to my IT people, go on showing me the faces of all those who are present here. And I have written to Goodwill that make a list of all, because all those who are sitting here are lucky persons to attend this lecture. I told already, which has been declared by Silvamurthy, of this first and second trimester is the best lecture I've heard. Generally, I work 3-4 things together, a laptop, telephone call, dictation. But today, a lot of people must be unhappy and annoyed that I did not take any call, I did not dictate, and totally concentrated to each word of Alok Dhawanji. Because each word was so valuable, so many new models, Assam University, University of Ahmedabad, bringing the industrialists, technocrats, startups, and give them the facility. These are all ideas, Alok Dhawanji, that we know something, but we have to do more in more emphasis. And all my scientists sitting over here, they are, I must tell you, brilliant. I always declare, bring me any other university in the country who has such scientists as my university, then I'll bow before them 5 times. But there is no. They are self-motivated. They are doing a lot of research. And Dr. Lamurthy, when we have completed 2,000 patents, then we will celebrate. At the moment, we have 1990 or something.
1940, Founder President.
1940. So when they are, the Smita Sahu should tell us. So my dear scientists, today, a lot of ideas have come. And he has rightly told he has made a fire. And what he told, we should उसको खत्म करें. No, fire तो अब आगे चलेगी. That one, I always take one matchstick, burns with one matchstick 1,000 diyas. So like that, your thoughts, what you're given today, has made at least my life more difficult, more challenging, because I've written 21 points, which I will be sharing with my people. They will be sharing with me. I will be writing to all those who are sitting here in one page. If you want, if you have time, write me in one page what was take home from this. If you feel comfortable, because just one page, I have heard it. I know about Alok Dhawanji, everything. I read the biodata, but I want to know, Sanjay Kumarji and everybody else, what you understood to take home. There are so many valuable points. I have never heard such innovative initiatives which a person like Alok Dhawanji has taken, and we should grasp it, we should implement it. And Dr. Naudi, I wrote you that it was a good topic, good speaker, and you invited the good people. So I think so many people came, very wise. And what we will do, those who could not come, Dr. Murthy,
Yes, sir.
at our university here in UP and all our other universities, we will deliberate, we will telecast this lecture again, and I will again attend it with all my persons. There I want 1,000% scientists from Thunder Force. The Thunder Force has 2,000 people, members from all our universities. So what we will do, we will arrange this telecast of this lecture, having 1,000 scientists. But there is so much to take home. There is so much to copy. There is so much to understand. And I think that my people are doing it. But there are new ideas. There were today many new ideas we should implement. And my again humble request to all those sitting over here, if you can, convenient to you, write one page only, what is the take home? Because there is so many take home. Alokji, actually I should have told Srivanti not to let you go back. Hold him here. Hold him here. And because you would never find such gentry sitting over here, because you must be lecturing anywhere, but these are exceptionally different people. Everyone is different. And if you sometime find other university where you feel there were equally brilliant, self-motivated scientists, then tell me, then I would like to visit that, but there is none. Thank you very much, Alokji. It was a honor.
Thank you for your blessing, sir. And as you rightly mentioned, I think each one of them is very distinguished in the gathering, both on the dais and off the dais. And they've been handpicked by you through your colleagues. They've been contributing. I've seen amity grow, as I did mention in my early part of the talk. Both the Noida campus and the Lucknow campus, that how they've grown not only in the terms of infrastructure, but the stature and the quality of research and the quality of students that are now coming out of the university. So I think my salutations to you, sir, that you've been able to change the way education is imparted in the private sector. And I think more universities that are coming up now are following this model. Currently, they're limited to maybe one, a maximum of 2 campuses. But the fact that you've been able to get, I think, 11 or 12 campuses, not only across the country, but across the world, is again remarkable. And I feel that we did start a course at Ahmedabad University with Judge Business School, where the students who were pursuing family business MBA, they could actually spend the last month at Judge Business School, and they would take the project with them, and they would actually discuss with the topmost leaders in terms of academics in the business school, and tweak the project so that they could implement it in their businesses. So I think that was a very hands-on approach, especially in the Gujarat region, because that's where you want to see how you can transform the community. And
Right.
the other thing which I could mention here, sir, is what is the impact in 1 square mile, and that is how institutions are judged. So if you've been able to make a difference in the 1 square mile where any of your campuses are, I think that should be documented, because those people then become your ambassadors forever. Because of this university, this transformed. So let's say because of the university in Noida, did the economy transform? Maybe the social scientists in the university should document it, where it was before and what happened after and what will happen 10 years from now. So I think series of these documentations, if they can be done. So university townships are known in the world, sir, that once a university develops, the entire township develops around it. I think that's how they gauge the societal impact. So one is, of course, the intellectual impact that the universities bring or institutions bring, and the other is the society impact or societal impact that the institutions bring. And one instance I'll share, and then I'll stop. That the Honorable Minister of Science Technology, Dr. Harshvardhan, was taking in, presentations from all directors of CSIR, and he mentioned that I just want to see 5 slides, and those templates were sent to us. One of them was societal impact or impact, right? So because of Toxicology Institute, we don't have direct economic impact, but the societal impact based on especially the initiatives that were taken in food was assessed by a third party, and it turned out to be 1,000 crores per annum for the rest of the life of the institution. And at that time, our budget was only 60 crores.
Right.
Immediately, he took a picture of that slide and he tweeted it, that this is how it should be. So it's not only direct ROI, which is in terms of money, but indirectly how we're impacting the society. I think that's equally important for an institution. So thank you once again for your valuable time and being there for my lecture. I really appreciate that.
Dr. Alokji, in addition to the scientific deliberations, you also showed many leadership styles, examples. Nowadays, I'm very busy to develop a ideal leadership style globally. And therefore, you have these birds, those who snatch coming down, and these all can be made in a leadership book because this the bird leading, and then if she get tired, then less joins. So I think these leadership, what you showed, will come in my book. I hope you don't have the copyright for that, because, having such people in my 11, 12 universities, and now Dr. Sulmati, once we should take Dr. Alok Dhawan to the Global Research Hub, New York, Long Island, and also to our Dubai campus, which is developing the best university in Emirates, so that when we do it in New York, then a lot of scientists will call from all over the world online or physically from Canada or USA, because there are ideas that everybody would like to listen. So I think once again, thank you very much and compliment to all. I think you are equally unrestful as me after listening this lecture, because वो कहते हैं ना, जब वो राजीव शर्मा जी कहते थे अभी तो पार्टी शुरू हुई है, अभी तो पार्टी शुरू हुई है, तो वही मैं कहूँगा अभी तो पार्टी शुरू हुई है. Thank you to everybody.
Thank you, Founder President. Thank you very much. Now, I would also request briefly one minute Dr. Chakravarti, Dr. A.K. Singh, and finally our Sanjay Kumar ji to make some remark and conclude this. Over to you, Dr. Chakravarti.
Now, when I left home for the office today, I thought that I think it is a very special day. And when I met Dr. Dhawan, I think we have been walking together at CSIR for quite some time. I think I made my day. And the lecture was really fascinating. He has been a very good photographer in CSIR. That's why his photos on the eagles were so beautiful. But in CSIR, we also used to call him, this is a man who has an eagle's eye. And that's why when he titled his talk, Supporting Risk in Science, I remember that was Dr. Mashelka's time when he started this crazy idea fund. The ideas which are crazy, nobody will fund in the world. No DST, no debatable fund, crazy ideas. But he created that fund, crazy idea fund. That means from that time, supporting risk in science started. And he was instrumental. And thank you, Dr. Dhawan, for this stimulating talk. And one more point, he is not only a very distinguished scientist, he is an institution builder. He builds institution, he creates institutions, and through that, he makes the national impact. Thank you very much, Dr. Dhawan, for coming to Amity.
Okay, I think due to paucity of time, I'll be as brief as possible. I think excellent talk, and I think I'm not finding word more excellent articulation. You know, the way you have conveyed your content, I think unparalleled, mesmerizing. And I think the idea which you had been driving, the risk support, I think not only me, I think all of us wouldn't agree more than that, you know, once it is supported and there is a risk taker, I think it is a deadly combination. The outcome can only be one, that is success. And, you know, I would seek the, you know, response later on, because when you say the toxicity, is there any trend or correlation between the molecular form and the nano form in most of the cases? Does the toxicity always enhances, or in some times it doesn't enhance, it becomes beneficial? You know, I am not asking your response. Now, a second generation scientist, I think though his father had been a stalwart, and I don't know whether I am authorized to say that or not, I think certainly he is going to outshine the personality of his father in times to come. Thank you very much.
Thank you, Dr. Sanjay. After that, Gopal will give the formal vote of thanks.
Very good afternoon. Indeed, such a great feast today we had with Dr. Alok. Both of us are together for last so many years. In fact, you know, even before becoming the director, we were together and we had known each other so well. And we joined directorship, I think we took almost together within a gap of 1 week or so. And that day onwards, we had several things together. And, you know, you must have noticed two things. One is that philosophy and science, when they are combined together, what a new things emerges. And in today's lecture, you must have noticed a lot of philosophies. And that's what PhD is all about. Whether he talked of sky versus sky, whether he talked of risk taking. And in the last, he mentioned something very interesting, although that you may do anything, you may discuss anything, but till you recommend and take it to the final end, that's very important. What he mentioned about this nanotechnology initiative in his, you know, in the country. And I must also mention, sir, he had taken several initiatives which are of national importance. And apart from science, he has several qualities, like the movie that you saw just now. And I think very few people will have that quality, that apart from doing good science, they also know how to project it to the general public. And that you saw in the form of that movie. Several of us might be very good scientists, but may not be that good communicators.
It's a God's great, you know, blessings.
That's right. That's right.
Thank you.
So, thank you, sir.
Before I go to Gopal for a formal vote of thanks, Goodwill, can you stand? She is the one who coordinated the whole event. Let's give a big clap to Dr. Goodwill Coker, the Deputy Director in ASTIF. Now over to you, Gopal.
Actually, everything has been said firstly by Founder President Dr. Salvamurthy and all the speakers on the days about Dr. Dhawan. My introduction to CDRI goes back when I was probably 9-10 years old. And last 1 week, we have a couple of interactions with CDRI scientists. I think last week we had Dr. Srivastav, Dr. S.K. Dubey,
who spoke about skill development. And now so my older brother, it's a cousin basically, Dr. R.C. Gupta, who worked in CDI for many years. And Dr. Nityanand's name was a household name, followed by Dr. Dhawan, Dr. C.M. Gupta. So that's my introduction to CDI. We used to go to CDI in the evening when we were very young. My brother used to take me to experiments. But I am not that qualified because I'm not a biochemist to comment. But whatever has been presented by Dr. Dhawan is very motivating. And with all you are present here, I think we should take advantage of his coming here and now his offer to work together. So we are very thankful to Dr. Dhawan for his acceptance and visit us and giving a very innovative talk on a very topical subject. And this is 124th STF lecture and second for this year, and we'll continue with the series. So my thanks to all of you present here, and we look forward to have your participation with the institution which Dr. Dhawan heads. And my sincere thanks to Dr. Sulamurthy also, because he is the driver for all these innovation talks and selection of the speakers and the audience. Very thankful, very select speakers and audience to have a meaningful dialogue. So thank you very much. And nevertheless, my thanks to Dr. Goodwill. She has been she worked very hard to organize this, especially this talk, and inviting all of you personally, calling all of you. Thank you, all of you. I look forward to have.
Thank you. Thank you, Gopal. Incidentally, today is the 7th death anniversary of his father, Dr. 6th, the death anniversary of Dr. B.N. Dhawan, the father of our Dr. Alok Dhawan. So that's such a noble soul. We'll be blessing him right from the heaven, that, such a wonderful son he has given to the country, to the humanity. So let us pray for his soul to be happy wherever the soul is. And thank you, Dr. Dhawan, once again. And clap for every one of you who is here. Let's clap for each one of you. Thank you. Just 1 minute, please.
Very special photo.
कहीं भी हो, अलग धवन जी कहीं भी हो,